Hello and welcome to this lecture on cytokines, lecture number 1 of the cytokines. Now, we have seen in previous classes that many of the reactions of the immune system are dependent upon recognition of antigen. Now, just like hormones in the field of endocrinology are secreted in order to adapt to newer situations or changed situations, hormones are released which enable the body to react and establish a new equilibrium with the new set of conditions. Basically, hormones alter the way different kinds of cells metabolize and react to the new situations by coming up with newer and adaptive responses. Similarly, the immune system faces newer and altered situations when it is faced with incoming infections. And apart from the specificity of the immune system that is mediated by the molecules such as the T cell receptor, the B cell receptor, molecules of the adaptive system, adaptive and innate immune system, the cytokines are the ones that are similar to hormones in endocrinology that help the cells of the immune system to adapt to these changed situations. Basically, then, what are cytokines and how do they function? Early on, after it was realized that reactions such as the mixed lymphocyte reactions, in which two populations of lymphocytes recognize each other as non-self, which result in the proliferation of cells, immunologists knew that this was the end result of the activation of lymphocytes, but they did not know what mediated this activation of proliferation that usually resulted in such allo responses. Now, we know of course, the first signal comes through the T cell receptor, and then of course, you have the co-stimulatory signal. Then you have to have subsequent signals which have to allow the T cells to proliferate, which means changes in the phases of the cell cycle. Normally, T cells or naive T cells or unexposed T cells that are unexposed to antigen and not active, they are normally in the T0 or the G0 phase, which means that they are restive in the restive state. In such situations, lymphokines or cytokines or growth factors cannot act on these lymphocytes, because they would not have or they are not properly receptive to the availability of these lymphokines. It is only after the exposure to the antigen and the signals that ensue through the T cell receptor and the co-stimulatory signals do the T lymphocytes put out on their surface receptors that could engage the cytokines that are available in the media. So, these cytokines were first discovered in, as I was mentioning, in alloreactive MLR situations, where they found that the supernatant of allo mixed lymphocyte reactions had in their supernatant growth factors that enabled these lymphocytes to proliferate in response to the availability of those lymphokines. As shown in this slide, you find that cytokines may be looked upon or now known to be of low molecular weight soluble factors, which are secreted by cells in order to communicate with other cells in the vicinity, mostly in the vicinity. Some cytokines can function similar to hormones and act on cells in a distant location. Now, this ability to communicate with other cells in the vicinity normally or always occurs by binding to high affinity specific receptors. In other words, most of these cytokines always bind to high affinity specific receptors, and the end result is the modulation of the function of these T cells. So, looking at the history of allo responses, it was first discovered that the supernatant obtained from an allo mixed lymphocyte reaction or responding lymphocytes. In other words, when you mix two strains of lymphocytes together and allow them to be cultured for a period of 3 to 5 days, these lymphocytes recognize each other as non-self and they get activated. This process of activation releases certain growth factors into the supernatant. So, what was done was to remove the cells by spinning down the cells and take the cell free supernatant and add it to normal cells that have been exposed to the antigen, and it was found that this supernatant allowed the cells to undergo proliferation in a much better way. So, they had this supernatant had the ability to support the proliferation of T cells that had been activated. So, this was initially called as the T cell growth factor, which is of course today's IL-2. So, looking at such factors, one needed to culture large amounts of large numbers of lymphocytes and to purify large numbers of allo supernatant obtained from alloreactive MLR reactions. Of course, later on the ability to clone DNA and recombinant DNA technology enabled the cloning of these genes, and now of course, many of these cytokines are available as recombinant DNA or recombinant DNA that can be put into various kind of vector systems, expression vectors, to make them express and to purify these different lymphokines in large amounts. At that time, basically the limitation in the number of cells that one could utilize in antigen specific mediated immune responses made it impossible for one to purify the large number of lymphokines that are known today, which then of course became possible because of recombinant DNA cloning. It was Cohen in 1974 who referred to them as chemical messengers or communicators between cells. Most were growth or differentiation factors, and they would regulate embryogenesis, hematopoiesis, tissue repair, inflammation and immune function. That is, processes that are involved with normal growth and development involving healing and repair. So, all this was supposed to encompass all the different reactions not only of the immune system, but of various other kinds of processes in the body. They were subsequently discovered, the cytokines were subsequently discovered to be proteins, more so glycoproteins. They were soluble in nature, low in molecular weight, about 15 to 25 kilodaltons, and they would be available only in very low concentrations. And they were stimulus responsive. Stimulus responsive meaning that when you stimulate a T cell or a leukocyte, this would end up secreting the different kinds of lymphokines. In other words, inactive or unactivated or cells that were not exposed to different kinds of stimuli would not secrete these lymphokines. So, therefore, the secretion of these lymphokines had to be inducible. In other words, these were not constitutive except for a few cytokines. Most of these cytokines are inducible in nature, inducible meaning that they were stimulus responsive. The availability of specific receptors made the action of these cytokines specific in nature, and many of these cytokines were found to be multifunctional, and they were acting at short distances. In other words, this was something of a feature that would regulate the action of these lymphokines whose action would be very potent otherwise if they were could stick around for long periods of time. One of the features that enable specificity of action of these lymphocytes of these lymphokines is their short half life. For example, a lymphokine known as tumor necrosis factor has a very short half life of about 15 minutes in vivo. Secondly, these lymphokines are usually secreted in the vicinity of the interacting lymphocytes. So, all of us now know that T cells recognize antigen in association with the MHC complex that is presented or available on the surface of antigen presenting cells. Hence, and cell to cell contact is a must in many of these reactions. This cell to cell contact enables the formation of minute microscopic pores or so called antigen bridges through which these lymphokines can get across to the cell that had to be stimulated, and therefore, the proximity of location made the possibility of a strong active cytokine in that location. They did not have to diffuse for long distances, and these are essentially unstable molecules subject to degradation over long distances. These cytokines have the ability to not only to act by themselves, but they also can synergize and they can antagonize each other's function. In other words, this synergy or antagonism is what sets up the balance of the immune system that we finally see as being inflammatory responses or overreactive Th responses that end up in autoimmunity, or too little a response that ends up in immune suppression. Most of these cytokines act together. In other words, in an immune response, it is just not one cytokine that plays a role. During the orchestration of immune responses, multitude of different cell types play a role, and therefore, because there are a number of cell types involved, each one of these cell types may respond by secreting or respond to by binding to these cytokines by binding them. There has to be a multitude or a network of cytokines that sets up this balance. This balance is actually between proliferation of cells or allowing T cells, naive T cells that have been activated to proliferate. Therefore, you have the specificity of action towards a particular antigen. In other words, antigen mediated proliferation of T cell responses to orchestrate T cell help, and B cell proliferation to a particular epitope helps it to make antibodies to that particular epitope. In order to make antibodies, these B cells have to differentiate into plasma cells, and more the number of plasma cells, the more the number of antibody molecules in the circulation. Therefore, these lymphocytes are the ones that are actually managing all these kinds of different proliferative responses as well as differentiative responses. For example, the differentiation of an active B cell to become a plasma cell that spews out or secretes large quantities of the antibodies that it should. So, they activate, as I told you earlier, many different kinds of cell types. So, going on, as I told you, basically then the specificity of lymphocytic response or an immune response is endowed by the T cells or the cells that are going to make the cytokines. Basically then, if it is a T cell, you have the incoming stimulus, which is the antigen recognition, which in the case of a B cell, it is through the BCR or the B cell immunoglobulin receptor, and in the case of the T cell, it is because of the T cell receptor mediated antigen recognition. This T cell then goes on to further phases of the cell cycle, which then allows it to respond to various kinds of stimuli or for that matter cytokines, and then spews out different kinds of cytokines by itself. So, if it is an antigen presenting cell, a macrophage, it would respond to TLR activation, or for that matter activation through various kinds of cytokines. They themselves will secrete various kinds of cytokines such as IL-1 or for that matter interferon. These cytokines that are then coming out around the vicinity of the activated cells, they are denoted here by these black spots, will go and then bind to the target cell. The target cells also can be of different cell types. So, for example, IL-2 acts mostly on T cells, although it can act on other cell types as well. So, this cytokine binds to a receptor that is expressed on these T cells, which are expressing a T cell specific receptor, which can bind to the IL-2 with high affinity. Now, this response can of course be modulated by modulating the number of molecules of this receptor, as well as the affinity of this receptor that we will come to a little later on. So, how do these cytokines avoid non-specific activation of T cells in an immune system? Basically, a naive T cell or a naive cell type that needs to respond does not have receptors if it is not activated. So, there are no receptors on the cell surface in an inactive or inactive T cell or a naive T cell. So, once the T cell is activated, the receptors are expressed on the cell surface and start to bind the relevant cytokines. So, you can see that the non-specificity of different kinds of lymphokines is avoided, because inactive or naive T cells or naive cell types do not express a particular receptor on the cell surface, and they are able to respond to these lymphokines only after activation and expression of the receptor on the cell surface. Going on further, the nature, what are the properties of cytokines is given in this slide. As I told you earlier, they are stimulus responsive. Stimulus responsive meaning that they are inducible by many stimuli, including the antigen via the antigen receptors. Other stimuli include those such as I told you, pathogens through the TLRs, or their components such as lipopolysaccharide, again through the TLRs, immunological stimuli and other kinds of cytokines. Now, these cytokines act in three different kinds of ways. They can be autocrine, meaning that in an autocrine situation, the cell that is secreting this lymphokine has the lymphokine acting on itself. So, these lymphokines act on the same cell that secretes it. So, this is called as autocrine. So, a T cell that secretes IL-2 can bind the IL-2 that it has secreted immediately and respond to it by activating various kinds of genes. As opposed to autocrine, paracrine is a situation where the cell that secretes the cytokine is not activated by the cytokine itself, but the cytokine goes and binds to another cell in the vicinity, as in this case, which is called as paracrine. So, depending upon the stability of the lymphokine, you have cells in the vicinity or at various distances from the secreting cell act as target cells. In addition to paracrine, of course, you have certain lymphokines that may act in an endocrine manner, which means that they bind like hormones, they are secreted in one location or made in one location, and then they go and bind to cells and bring about their action at distances away from the cell that actually secretes the hormone. So, here The cytokines that act in a endocrine manner, they bind to cells that are far away. For example, IL-1 is the endogenous mediator of fever. So, when you have fever, IL-1 is active. How does it do that? It comes from the tissue site where it is secreted, goes to the hypothalamus to regulate the body temperature. IL-6 acts at the liver, although it is secreted by different kinds of cells which are at different locations apart from the liver. They act on the liver to stimulate the acute phase response in response to different kinds of stresses. It also acts on erythropoietin, secretion of erythropoietin from the kidney. Now, erythropoiesis from the bone marrow is also stimulated by this sort of endocrine action of these kinds of cytokines. Cytokines are not produced in isolation in vivo. In vitro, of course, you can have cells producing certain kinds of cytokines, but in vivo they always act as a network. So, they are not produced in isolation and they are always acting together. It is the sum total of a variety of cytokines that produces a function. First, during the history, they were named after their functions. So, they knew what the functions of these various cytokines, and they were named after that. For example, colony stimulating factor, which stimulates the total number of colonies for the purpose of hematopoiesis. T cell growth factor, because this factor was purified from a mixture of T cells that were undergoing alloreactive responses. Macrophage activating factor, because you can have cytokines activating macrophages, activating the process of phagocytosis or opsonization. Now, the name cytokine encompasses the other terms such as lymphokines, which were given to those kinds of proteins or polypeptides that were secreted from lymphocytes, or monokines for those kinds of polypeptides that were secreted from activated macrophages and monocytes. So, all these different kinds of terms came under the name or now referred to as generally as cytokines, including those called as chemokines. As I told you earlier, most of them are inducible in nature, but some cytokines such as IL-6, granulocyte colony stimulating factor, and the other kinds of colony stimulating factors are mostly constitutive in nature, because they need to support hematopoiesis. Going on further, as they were discovered, the functions were discovered, cytokines can also be classified based upon their biological action. For example, mediators and regulators of innate immunity. There are cytokines that take part in innate immunity, those that take part in adaptive immunity, and those that they may have there might be an overlapping action between innate and acquired immunity. So, in innate immunity, activated macrophages and natural killer cells, NK standing for natural killer cells, secrete these cytokines in response to microbial infection. For example, IL-1, TNF alpha, TNF standing for tumor necrosis factor. There are two types of TNFs, alpha and beta, TNF alpha, IL-6 and interferon alpha and beta. There are many different types of interferons. Alpha and beta interferons are called as type 1 interferons, as opposed to immune interferon or gamma interferon, which is the interferon that results due to activation of lymphocytes, which then has different kinds of actions on the immune cells. These cytokines, they act mainly on endothelial cells apart from other kinds of cells, endothelial cells and leukocytes, to stimulate the early inflammatory responses. So, those cytokines that are liberated or secreted in response to TLR stimulation, such as TNF and other cytokines, they have action on endothelial cells, causing alterations in adhesion. Therefore, different kinds of lymphokines are trafficking into the different locations by altering the adhesion molecules such as VCAM, ICAM and so on and so forth. The mediators and regulators of acquired immunity are produced mainly by T lymphocytes in response to specific recognition of foreign antigens, via of course, the antigen presentation machinery. So, these include IL-2, IL-4, IL-5, IL-13 and so on, gamma interferon, TGF beta and TNF beta. Then of course, depending on the function, as I told you, because they are constitutive in nature, there could be stimulators of hematopoiesis. These are produced by the bone marrow stromal cells as well as leukocytes. They end up stimulating the growth and differentiation of leukocytes such as the stem cell factor, IL-3, IL-7 and GM-CSF. So, if you are to look at all these different kinds of cytokines, you can see that there is some overlap of MIP 1 alpha between the growth factors and inhibitors, and between, if you look at adaptive and innate immunity, you find that IL-18, IL-12 and gamma interferon are in the overlap between adaptive immunity and innate immune cytokines. These of course, in the green are those that help in innate immunity. The ones in blue here are helping with adaptive immunity, and there are various inhibitors to regulate the action of these various kinds of cytokines. IL-10 is mainly immunosuppressive cytokine, IL-1 receptor antagonist, TGF beta as well as soluble TNF receptor alpha. So, you have these cytokines which are constitutive, which are they act like growth factors. So, the action of these cytokines further can be described as being pleiotropic. Pleiotropic meaning that they can engage in different effects on different kinds of targets. So, a single cytokine has different actions on different targets. For example, IL-4 on B cells can have the action of activation, proliferation as well as differentiation. So, not only do they make the B cells proliferate, they also allow them to differentiate and allow class switching, meaning that class switching from one class of immunoglobulin molecules, they go on after switching to secrete a different class of immunoglobulin molecules, basically because of the gene rearrangement system that is already that we have already learnt about. On thymocytes and mast cells, they can act as a proliferative cytokine. So, you see they can differentiate B cells. On thymocytes and mast cells, they act they increase proliferation. The actions of many of these cytokines are redundant in nature, that means their functions overlap. This overlap is basically because of the property or the structural features of their specific receptors and the subunits that they encompass. They can share various kinds of subunits, and therefore, you have some of these functions overlapping between different kinds of cytokines, which we will learn in the next class. They have similar functions. Example, IL-2, IL-4 and IL-5 make B cells proliferate. So, they all have proliferative actions. Another example is that IL-4 and IL-2, they can make certain kinds of cells called as CTLL-2 to proliferate. These CTLL-2 was actually used to measure in a bioassay for the presence of IL-2 or IL-4, and basically it is a cytotoxic T lymphocyte that has retained its specificity for the lymphokine, but lost its specificity for the stimulating antigen. These lymphokines can be synergic in action. In other words, the action of two cytokines could be synergistic, that means more than additive. So, if the mechanisms are different, one would say that certain equal concentrations of two different cytokines will give you a doubling of its action, but in many of these cytokine actions are actually synergistic. So, more than additive in nature. So, IL-4 and IL-5 synergize to induce IgE class switching. So, in other words, IL-4 and IL-5 on their own would be stimulating a certain amount of IgE class switching, but when they come together it goes up many fold. Cytokines can be antagonistic. One cytokine can inhibit and alter the action of the other, which we learn much more when we look at T helper 1 and T helper 2 actions and how they differentiate. Gamma interferon, in another example, inhibits IgE class switching that is caused by IL-4 in activated B cells. So, B cells that class switch to IgE because IL-4 has bound to its receptor can be inhibited from doing so by the binding of gamma interferon to its specific receptor. TNF and TGF beta, they are antagonistic. EGF and TGF alpha are antagonistic. CSF and IFN type 1 interferons alpha beta are referred to as type 1 interferons, gamma interferon is referred to as immune or type 2 interferons. In addition to its functional classification into innate immune cytokines and adaptive immune cytokines, there are 4 major families of these cytokines based upon their structure and other criteria, such as interferon, TNF, chemokines and the hematopoietin family, which have a prevalence of alpha helical structure and little or no beta peated sheet. So, if you look at the interferon family, basically interferon type 1 interferons, they are secreted from leukocytes and fibroblasts respectively, and they are basically antiviral in nature. Virus infection induces enzymes such as oligoadenylate synthetase, and interferons actually help in this action. Interferon gamma, which is the immune interferon, they are secreted from Th1 cells, Tc cells or cytotoxic T cells and NK cells. They act on macrophages, proliferating B cells and Th2 cells and other cell types. They activate macrophages, they induce MHC both classes, both MHC 1 and 2, and they induce a class switch to IgG2A in differentiating lymphocytes. They block IL-4 induced IgE class switch as mentioned earlier, and they block, basically they are anti-proliferative in nature. TGF beta are produced from platelets, macrophages, lymphocytes and mast cells. They are basically chemotactic in nature, and they induce IL-1 from macrophages. Also they induce IgA switch in B cells. They inhibit the proliferation and promote wound healing in epithelial cells, endothelial as well as hematopoietic cells. Oncostatin M or OSM, they are secreted from macrophages and T cells. They inhibit the growth of tumors and induce the acute phase response. In addition to this, another cytokine that one comes across in different kinds of textbook is the leukemia inhibitory factor, which is secreted from thymic epithelial cells TEC, thymic epithelial cells during the differentiation in the thymus as well as bone marrow stromal cells. They induce the acute phase response and they help embryonic stem cells to proliferate. The other major family being the tumor necrosis factor is a very good example of a pleiotropic lymphokine. In fact, this is also referred to as a pro-inflammatory type of lymphokine, and these sort of TNF lymphokines abound in different kinds of autoimmune inflammatory situations like arthritis and so on. They are pro-inflammatory, as I just now mentioned to you, by the induction of IL-1, IL-6 and IL-8. These are the 3 different kinds of cytokines which are result in a pro-inflammatory situation which the TNF induces. They induce MHC1 on the cell surface of different kinds of cells, the gamma interferon receptor as well as the secretion of type 1 interferons. They induce what is called as antibody dependent cell mediated cytotoxicity, because they are activating macrophages, therefore, the induction of Fc receptors and B cell proliferation. So, they act on endothelial cells basically to increase adhesion molecules. So, lymphocyte trafficking can be altered, and they also act to increase the secretion of prostaglandins and the platelet derived growth factors. Basically, and all importantly, from the point of view of studying the mechanism of action of different kinds of cytokines, TNF is a very important lymphokine that induces apoptosis in cells. And of course, cachexia, depending upon whether it is TNF alpha or TNF beta. They inhibit lipoprotein lipases and have various kinds of other kinds of actions, such as being anti-tumorigenic and anti-parasitic in nature. The chemokine family are a very important set of polypeptides, which is a large family. They are small structurally homologous polypeptides, basically involved in regulating leukocyte migration and trafficking into different kinds of lymph nodes or into different locations within the body, like between mold tissues and other kinds of locations in the body. They are produced from leukocytes, epithelial cells, fibroblasts and endothelial cells upon stimulation with various kinds of lymphokines such as TNF, IL-1 or for that matter TLR mediated stimulation by microbes or antigen. They are usually small in molecular weight, 8 to 12 kilodaltons. They are structurally homologous and basically they contain 2 disulfide bonds or 2 disulfide linkages or 4 cysteine residues. These 2 N-terminal cysteine residues that participate in disulfide bonding form the basis of the classification of subfamilies of these chemokines. So, if these 2 N-terminal cysteines are adjacent to each other, they are called as CC chemokines. If they are separated by one amino acid, they are called CXC chemokines, where X is the intervening amino acid residue, which can be any amino acid residue, or they are separated by 2 amino acids, which is called as the CXX or the CXXX chemokines. They lack 2 cysteines in lymphotactin. Now, cytokines, the work on cytokines involves looking at the gene sequence. It has to be shown to be unique in order to be classified as a separate lymphokine. Their gene has to be cloned. So, the cDNA has to be available and the recombinant proteins must be shown to have the same activity as the native purified molecule in order to be named as a lymphokine. There are several different kinds of lymphokines, or they are called as interleukins, and these interleukins number from 1 to 35 and more. So, basically this lecture will cover some of these lymphokines, some of the important lymphokines, but for a more exacting or a more detailed treatment, one has to go into different kinds of textbooks, because the subject of cytokines or interleukins itself can go on for more than or several classes. Now, IL-1 is produced by most nucleated cells. They have a wide range of activities on many different kinds of target cell types. They induce hypotension, fever, weight loss, neutropenia and acute phase responses. IL-1, as I told you, can also act as a hormone in mediating in the fever response. As a dendritic cell derived factor, in other words derived from antigen presenting cells, it promotes T cell proliferation. In fact, this IL-1 secretion induces T cells to produce IL-2, which is one of the first responses that one sees in T cell mediated responses. As little as 10 molecules can induce IL-2 from CD4 cells. IL-2 of course, is a T cell derived cytokine, first named as TCGF. It stimulates the growth and differentiation of T cells, B cells, NK cells, monocytes, macrophages and oligodendrocytes. In fact, IL-2 is one of the cytokines that has been worked on very thoroughly by in the immune system. IL-3 is produced from T helper cells, natural killer cells and mast cells. It facilitates histamine secretion from mast cells. It is a hematopoietic growth factor which stimulates colony formation of erythroid, megakaryocyte, neutrophils, eosinophils, basophils, mast cells and monocyte lineage type of cells. IL-4, it is secreted from Th2 cells and basically it acts on B cells. It can also act on T cells to cause proliferation. It acts on endothelial cells, fibroblasts and thymocytes. It induces a secretion of IgE and IgG4 by B cells. IL-5 is a Th2 derived cytokine. It is also mast cell derived glycoprotein, stimulates colony formation from eosinophils, and importantly it induces IgA class switch in activated B cells. IL-6 is multifunctional. It regulates B cell function, hematopoiesis and the acute phase response. It is secreted by lymphoid, non-lymphoid monocytes, macrophages, Th2 and bone marrow stromal cells. IL-7. IL-7 is secreted by bone marrow and thymic stromal cells. It acts on progenitor B cells and T cells. The main lymphocyte population in the thymus that responds to IL-7 are CD4 negative and CD8 negative. IL-7 also promotes growth and differentiation of mature T cells, and it induces secretion of IL-2 as well as the expression of the IL-2 receptor. IL-8 is actually a chemokine. It is an inflammatory cytokine produced by many cell types, which functions as a neutrophil, chemoattractant and activating factor, and is involved in many inflammatory situations. So, it is pro-inflammatory. It also attracts basophils and a subpopulation of lymphocytes. It is a potent angiogenic factor and induces adherence to endothelial cells. Then going on further, another important cytokine being IL-10, which is secreted by Th0 and Th2 types of subsets of CD4 lymphocytes. It blocks the activation of cytokine synthesis by Th1 lymphocytes, activated monocytes as well as natural killer cells. It stimulates or enhances the proliferation of B cells, thymocytes and mast cells, and cooperates with TGF beta to stimulate IgA production by human B cells. So, there are so many such actions of different kinds of lymphokines. Another important lymphokine being IL-12, which is involved in Th1 responses. It is important against intracellular pathogens because it mediates finally the activation or production of cytotoxic T killer cells. It induces interferon gamma production by T cells and NK cells and enhances NK and ADCC activity. It stimulates the production of NK LAC cells or lymphokine activated killer cells. So, you can have killer cells that are activated by different kinds of lymphokines. Basically, they have a killing action on their target cells. These LAC cells are the cells that were that are found to be very active against a variety of tumor cells as they arise. This stimulates the differentiation of Th1 cells, and along with IL-2 it induces cytotoxic T cell differentiation. And going on further, you have IL-18 and IL-17, which are important, very important cytokines. IL-17 produced from CD4 or T helper cells activates the production of inflammatory mediators such as GM-CSF, IL-1 beta and TNF alpha by synoviocytes in arthritis, which is basically an inflammatory situation. IL-18 is produced by activated macrophages, can induce Th2 type cytokines alone or can induce gamma interferon production and promotes Th1 immune responses in synergy with IL-12. Now, IL-20 is a cytokine that regulates participation of keratinocytes in inflammation. Then going on further, of course, there are several of them which are IL-10 family members, and if you look at other kinds of cytokines, there are basically up to 35 of them. You can go through them at your own time, and all these cytokines, as I mentioned, work in a kind of a network. All these pathways of networks are quite exhaustive and can be found in different kinds of textbook, as well as in different kinds of catalogs of different companies, biological companies which sell these cytokines, or different kinds of antibodies. So, you will see that Promega company may have a website that may have their own articles on different kinds of lymphokines, and these networks pathways are given, and I would encourage you to look at these different kinds of pathways or charts that are available at different websites, since going through these networks and to describe them is a kind of limiting in this 1 hour of lecture. Then going on from cytokines to their specific receptors. So, if you look at these receptors, there are different, again they can be categorized as different families. As I told you, each cytokine has its own receptors. So, if you look at all these different kinds of receptors to the different cytokine families, you have what are called as the Immunoglobulin Superfamily of receptors. You have class 1 cytokine receptor family, which is also known as hematopoietin receptors. Basically, this is the largest group which binds most of the cytokines in the immune and hematopoietin systems. Apart from class 1, you have the class 2 cytokine receptor family, which is basically the interferon receptors. Then you have the TNF receptor family, then you have the toll like receptor family, and then you have the chemokine receptor family. So, you have these broad receptor families that have been classified. So, looking at the class 1 cytokine receptor family of receptors, you find the hematopoietin receptor as basically made up of domains or portions of the receptor that have certain kinds of motifs. They have conserved cysteine motifs in these domains, and you have another motif called as the tryptophan serine unknown amino acid tryptophan serine motif. So, in their primary amino acid sequence, these are the motifs that are available in the cytokine receptor. Basically, they are dimers, and the ligands that bind to them are listed over here, basically being IL-2, IL-3, IL-4, 5, 6, 7, 9, 11, 12 and so on. Going on to the subfamily of this class 1 or hematopoietin family of receptors, you have several subfamilies based upon their structure and the way that the subunits are make up this receptor. So, you have what is called as the IL-2 receptor subfamily, which share a common gamma subunit. Basically, you have for IL-2 and IL-15, they have 3 subunits, and these 3 subunits are made up of the alpha, the beta as well as the gamma subunit. So, you see you have the IL-2 receptor has the IL-2 receptor alpha, which has a short intracytoplasmic tail, not enabling it to mediate transmembrane signaling after it binds to its lymphokine IL-2. You have the other 2 subunits having longer intracellular segments, which enable them to mediate signaling events below the membrane. That is what results in the IL-2 action on the genes that it has to activate. We will come to that in a little later on in the next cytokine class. So, basically if you look at this color, you see that all these different cytokines share this particular subunit, and they have of course the WS the XWS motif, and they have the conserved cysteine motifs. So, in addition to this common gamma subunit that is shared between IL-2, IL-15, IL-7, IL-9 and IL-4, the other subunits have their specific lymphokine binding subunit. For example, IL-15 shares the IL-2 receptor beta, but the alpha subunit binds to the IL-15 lymphokine, and the IL-7 receptor subunit alpha binds the IL-7, and so on. So, these have been classified together based upon the sharing of this common gamma subunit and called as the IL-2 receptor subfamily. Then you have the IL-6 receptor subfamily, which shares a common glycoprotein subunit of 130 kDa. So, you have these over here that are in IL-6 of course, you have 2 of these GP130 units, and then if you have the other lymphokine binding subunit, again characteristically having a short intracytoplasmic segment, basically saying that the GP130 mediates or able to mediate the transmembrane signaling subsequent to the binding of its lymphokine. So, you see IL-11 share the same GP130, but has a different IL-11 binding subunit. So on for the ciliary neurotrophic factor CNTF or the oncostatin M factor or the leukemia inhibitory factor. Then you have the GM-CSF receptor subfamily, which shares a common beta subunit as shown over here in this color. So, you have the receptor specific subunit, and then you have the longer intracytoplasmic segment, which enables the transmembrane signaling, which is the common beta subunit between GM-CSF, IL-3 and IL-5. So, then of course, going on to the class 2 family, this is not a subfamily, this is a separate family, which is called as the interferon subfamily, which has the different kinds of interferons. Only 3 of these are listed over here, alpha, beta and gamma, but there are several different other kinds of interferons that have been discovered. In addition to this, of course, you have all these other kinds of lymphokines, IL-10, which is a immunosuppressive cytokine, basically being a dimer and consisting of cysteine motifs. Then you have the TNF receptor family, as mentioned earlier in the classification, having this single subunit that basically has 3 motifs called as the C1, C2 and C3. Basically, they have TNF binding, they are TNF binding. So, TNF alpha, beta, and then you have the CD27 ligand, the nerve growth factor and the FAS antigen. So, then of course, you come to what is called as the immunoglobulin superfamily. What is the immunoglobulin superfamily? It is basically the disulfide bonded domain that is available in the immunoglobulin domains that are found, and many of these domains are shared between different kinds of molecules both in the immune system and outside the immune system. So, if you look at these molecules, you find of course, as you saw earlier, you have the membrane and then you have these immunoglobulin domains forming this subunit. So, these are the disulfide bonds which is characteristic of these immunoglobulin domains, and these are the immunoglobulin superfamily of receptors, and the ligands being IL-1, IL-18, MCSF and c-KIT. Going on further, you have the chemokine, I am sorry about this, this has to be the chemokine receptors and their ligands. You have the ligands being IL-8, RANTES and MIP1. So, in this case, these are different from the rest of the receptors in having transmembrane domains. So, they have 7 transmembrane domains. So, you have the receptor. So, you have something like this, and these receptors or the chemokine receptors are coupled under the membrane to G proteins. So, these are G protein coupled receptors, basically for the purpose of downstream signaling or the transmembrane signaling events subsequent to the binding of the chemokine to its chemokine ligand. So, basically then you see that looking at cytokines, they were first discovered in a fashion where they looked at supernatants of allo reactive lymphocytes. Subsequently, these different factors, the genes for these different factors were found and the receptors were characterized, and all these different kinds of lymphokines, the structure was worked out, and based on their structure and function they were classified into different families and superfamilies. So, all these different kinds of lymphokines bring about their action by binding to their receptors and are regulated in a very unique way. So, the way they are regulated actually has a lot of relationship with the different kinds of receptor families that we just now recognized, and that is having a common subunit. Basically, if you have a common subunit shared between different receptors, you can envisage a situation where a dearth or a limitation of this subunit can actually cause a limitation in the kind of lymphokine action, although there are different kinds of lymphokine binding subunits that are available. Even if more number of subunits are available for a particular lymphokine, if the common shared subunit is limiting in number, then you would find that there would be a limitation in the outcome of that lymphokine, although the lymphokine is produced in large amounts. For example, a very interesting situation arises when you have T cells proliferating. T cells secrete IL-2. When the activation is strong, you have more of this growth factor or more of IL-2 being synthesized in the medium, and therefore, one would think that the more the amount of IL-2, it should go on resulting in a continuous increase in the number of lymphocytes that are responding to this lymphokine IL-2. But what actually happens in vivo, or for that matter in vitro also, is that after a period of time these lymphocytes fail to respond to IL-2, even though large amounts of IL-2 are available in the surrounding medium. And this is a case of receptor downregulation or altering the affinity of that particular receptor. So, it does not respond to excess of lymphokine. In fact, this is how lymphokine responses are regulated, and we will come to that in the next class. So, this class has dealt with the various kinds of lymphokines, their structure and